Okay, good evening everyone. I guess people will keep on joining and we shall start the session because it's already 6 p.m. So let us start the session. Okay, so I hope my audio and video will be clear to everyone. Alright, so today it would be week 4 of NPTEL live session and the topics for today's discussion would be nanotechnology in point of care diagnostics, nanopharmacology and drug targeting. In addition to that, we'll also see cellular uptake mechanism of nanoparticles or nanomaterials, the various in vitro methods that are available to study the antibacterial and anti-cancer properties of the nanomaterials. There are different nanomaterials which are fabricated basically for different anti-cancer and antibacterial applications. In addition to that, we'll also see the nanotoxicology, which will basically be in which we'll basically be talking about how nanoparticles, some of the nanoparticles and some of the materials with which the nanoparticles are fabricated can lead to cell toxicity, and what are the different ways with which we can overcome that. So that is one of the downsides like of some of the nanoparticles. Now let us start with the session. So the first topic would be point of care diagnostics. So what exactly you understand from point of care diagnostics? So it basically provides great potential for detecting and monitoring infectious diseases, especially in remote areas where there are no large decentralized diagnostic facilities. So if you imagine, point of care diagnostics basically refers to fabrication of miniature kind of devices for different purposes. Like in this case, they are talking about diagnostics. So, as we all know that in distant locations or in basically when we are talking about remote locations where a lot of healthcare facilities are not available, uh, there are no large centralized diagnostic facilities, especially at remote locations. So, in those cases, what can be done is the research is being shifted, the research is being done on fabrication of these miniature devices that can be carried to such remote locations so that diagnostic testings can be done. So they also need to be robust enough to withstand extremes of temperature and humidity because obviously there would be that would be a low resource setting, villages, remote locations. So all those are low resource settings. So a lot of resources won't be available. Just a second. Okay. So POS diagnostics should be capable of analyzing small volumes and body fluids. So the diagnostics that we are talking about, the miniature devices that we are talking about, that shall be fabricated, it should be capable of analyzing small volumes of bodily fluids like blood, saliva, urine, etc., detecting a deadly chemical or microorganism with low concentration. So the sensitivity and reliability of the instrument should be such that we can basically rely on the results that they are giving, at least for the preliminary analysis. Now, for example, the detection of a very low concentration of dangerous bacteria in drinking water, because we all know that when we are talking about villages basically or any of the remote locations, whenever we're talking about remote locations or villages, there would be there would be lack of resources. So in that case, in those situations, in those scenarios, we need to come up with some technology so that we can go there, we can carry that we can carry those point of care diagnostic devices to those remote locations so that those people can also benefit from the technology. So after diagnostic testing with POS devices, the device should be disposable to protect the end users from biohazardous samples produced, because we'll be using those devices, those point of care diagnostics for analyzing some biohazardous samples. Now, coming again, in low resource settings, the budgets for healthcare facilities are extremely low. This is what I just now stated. To reduce POC test cost parameters, including minimal use of expensive reagents, inexpensive manufacturing for mass production, quality control, and miniaturization should be considered. Moreover, there is an extreme variable in different regions and seasons. Therefore, medical diagnostic devices, clinical use should not require external water and withstand large temperature fluctuations. So basically, they have highlighted what are the different, uh, I would say, drawbacks or shortcomings when you're working in low resource settings like remote locations or villages, because you won't be having, maybe you will not be able to store samples for very long time. You won't be having proper storage locations. You won't be having sophisticated instrumentation. So in those conditions, the diagnostic devices, the point of care diagnostics that you are fabricating here, it should be capable such that you are able to do the analysis in those extremes of situations as well. So this is one of the example, like as you can see, they have fabricated a point of care diagnostic device. All right. So this is basically a microfluidic device integrated by biosensors and smartphone to load blood, urine, or saliva samples. So suppose you are putting your samples here, it is a microfluidic device, there are microfluid channels. So what a sample you're collecting, suppose saliva. So it will move through here, and then here there would be biosensors that would be detecting some of the biomarkers of pancreatic cancer. So if your sample that you're putting here is like, if you are someone who is developing pancreatic cancer, there shall be some biomarkers. So those biomarkers would go and bind here, and based on that, we'll be getting some signal, some optical signal based on whatever interface you would have used and what kind of sensing you are applying. And this microfluidic device, as you can see, is being integrated with the smartphone. So can you see the miniaturization of the whole setup? So you'll get the, you know, kind of analysis kind the results. You'll be getting results for that. Like suppose if how many biomarkers for pancreatic cancers are present in that sample. So it can be used as a means of preliminary analysis to diagnose some of the diseases even at the early stages. So it can also be used for different routine checkups, routine diagnostics at remote locations or in villages where people don't have access to the healthcare infrastructure, healthcare facilities, and are often neglected because of obviously the economic thing, financial thing. Now, let us do some of the sample questions to see how much we have understood what exactly point of care diagnostics mean, what is the essence behind fabrication of small miniature devices, small microfluidic devices. So the first question is, point of care diagnostics are designed to provide results. So I'll wait for 10 seconds, 20 seconds, 10 seconds for each question, and then we'll discuss the answers of these questions. Read questions carefully. I kind of encourage students to write your answers in the chat box because it helps. Like this live session is basically for you people to practice. So try attempting the questions. Okay, so let us discuss the answers. So point of care diagnostics are designed to provide results. The correct answer is B1, because obviously point of care diagnostics are designed to provide results directly at or near the patient site, because of the obvious reasons, because the whole purpose of fabricating these miniature point of care diagnostic devices is to help people from distant locations, so that you can, like, making a miniaturized version so that you can carry it easily to off distant locations, to villages, and then you can do the testing then and there only. So the correct option would obviously be directly at or near the patient site. The second question is, which is a classic example of a point of care diagnostic test? The answer would obviously be pregnancy kits are simple lateral flow point of care devices. So you all must be knowing about the pregnancy kits. It's a small kit in which you put your sample here, and based on whether you have human chorionic gonadotropin, which is one of the hormones, you get lines. One line, two line. We also discussed about lateral flow devices in our earlier sessions. So pregnancy kits is one of the classic example, one of the simplest example of what exactly point of care diagnostic means. So the correct answer is lateral flow pregnancy test. So the correct answer is lateral flow pregnancy test. Moving forward, let us understand how exactly pregnancy tests work. So what is exactly we are detecting in a pregnancy test? Pregnancy test detects a hormone called human chorionic gonadotropin. This hormone is produced by the placenta from the time at which the embryo attaches to the uterus. So HCG is essential for the function of the corpus luteum, a temporary structure in the ovaries that produces the hormones progesterone and estrogen. It has been linked to early pregnancy symptoms. So this particular hormone, this human chorionic gonadotropin, has been linked to early pregnancy symptoms such as nausea and vomiting. HCG is eliminated in urine and can be detected by pregnancy test around 9 days after fertilization. So once the fertilization has taken place, then after 9 days, you can detect HCG from where? In urine. So what you do? This is a sample pad, you put your sample here. So this is a drop of urine. Then there would be some antibodies that would bind to some of the agents present in your sample. So urine is applied to the sample pad. If a woman is pregnant, urine contains HCG. Okay. Then HCG binds to mobile antibodies. These antibodies also have an enzyme attached to them. Immobilized antibodies in the test zone bind to HCG. The enzyme on the first antibody changes the test line color. Excess antibodies bind to immobilized antibodies in the control zone to show the test worked. So, as we have also discussed in our previous lectures how lateral flow devices work, there will always be a control line. So, if the patient tests positive for pregnancy, there would be two lines here that would be coming. Otherwise, there would be only a single line, which would be a control line. So, pregnant, and then this one control line means non-pregnant. So, this is how miniature point of care diagnostic devices are being fabricated uh, and being used in nanotechnology. Now, let us do some more questions. The second question is, a major advantage of point of care diagnostics is, and the correct option. So try attempting these questions. I'll just wait for 10 seconds for you to, at least if you're not like writing in the chat box, I would suggest you to at least do it in your mind. Like, what would be the correct answer? I'll wait for 10 seconds. So the first question is very obvious. The first question is very obvious. A major advantage of POC diagnostics is Obviously, the answer would be C, rapid and user-friendly results. Obviously, you can do the diagnosis then and there only, so it is rapid and user-friendly. The second one is the main challenge of point of care diagnostics in healthcare is the correct answer would be POC test may have like accuracy and sensitivity compared to lab tests. So obviously, whenever I'm talking about point of diagnostics, okay, so keep this in mind that it is it is only for preliminary testing. Okay, you cannot completely rely on the test result obtained via point of care diagnostics because it has lower sensitivity and specificity in comparison to laboratory methods. Point of care diagnostics is being marketed, is being researched upon because at least for the preliminary testing to uh, that at least for the preliminary testing you can use this point of care diagnostics. Now moving forward to the main topic of today's discussion. In last few lectures, we discussed how, we synthesize, we fabricate different nanoparticles, load it with different drugs, different therapeutic agents, and do the delivery. Now, in order for those nanoparticles to be effective, it is very important that the cancer cells take up those nanoparticles. So today we'll be talking about the different uptake mechanism of nanoparticles. So cellular uptake is a critical factor in the effectiveness of nanoparticles for biomedical applications such as drug delivery, imaging, or therapy. Efficient uptake ensures that nanoparticles can enter target cells, interact with intracellular components, and release their payload where needed. So, an efficient uptake is one. Suppose you have cancer cells. Suppose, okay, you have different cancer cells. This is the nucleus. Now, suppose you have treated these cells with the nanoparticles that you have synthesized. Now, in order for these cells to be killed, it is important that these nanoparticles go inside the cells by some or the other mechanism. So this is what we'll be talking today, that what are the different mechanisms that are used for cellular uptake. Now, one important thing to consider here is until and unless these nanoparticles, these nanoparticles are the ones which are carrying the drugs. Okay. Now, in order for the cells to be killed, it is important that the drugs or the therapeutics that has been loaded in the nanoparticles are released into the cell because that will only lead to cell death. So, the mechanism and rate of uptake influence the nanoparticles distribution, therapeutic efficacy, and potential toxicity. Understanding and controlling cellular uptake allows researchers to optimize nanoparticles design, size, shape, surface chemistry. Now, based on whichever approach you are targeting. So suppose you are trying to make a hydrophobic nanoparticle. You made a hydrophobic and you took some nano carrier and made nanoparticle out of it, and suppose it is hydrophobic in nature. Now in this you loaded some now in this suppose you loaded some of the drug molecules. It has drugs in it. Now if it is hydrophobic in nature, it can penetrate to the plasma membrane. But in that case also, it can go in by endocytosis. But again, there are different mechanisms, like different types of endocytosis, which will depend on uh, its structure that through which it is being made. It was made, what was the material, and how it is being internalized. Moving forward, there are different cellular there are different mechanisms through which nanoparticles can make cellular entry. The first one is clathrin dependent, caveolin dependent, clathrin and caveolin independent means there is no clathrin or caveolin involved. Then phagocytosis and micropinocytosis. Now, phagocytosis and pinocytosis are the two terms that you might have heard in your previous classes. Like this one was mainly popular as eating, and this one as cell drinking. So, this one is a schematic overview of nanoparticle uptake pathways via endocytosis. Multiple different pathways exist for cellular entry of nanoparticles via endocytosis mechanisms. The first one is clathrin dependent caveolin. All the names that I have just taken now, you can see it here. These nanoparticle cell uptake pathways are mechanistically distinct and highly regulated. Now, one thing to consider here is this particular line that all these nanoparticle cell uptake pathways are mechanically distinct and highly regulated. So might two nanoparticles with some differences might not be able to get internalized in cell because with the via the same pathway. So the pathway would be different. So it's not necessary that if a particle is of 5 nanometer, okay. Suppose there are two particles, one is of 5 nanometer and the other one, let's suppose the other one is also 5 nanometer. But there might be possibility based on the nature of these particles that which mechanism they will utilize to get inside the cell. So always remember this. Now talking about the different types of cellular and I mean cellular uptake mechanisms. So these are broadly divided into two categories. The first one is endocytic mechanisms. Okay, endocytic mechanisms means some type of endocytosis is happening, which is pulling the nanoparticles inside the cell. And the second one is non-endocytic mechanisms. Okay, we'll deal each of these one by one. The first one is phagocytosis, recognition of nanoparticles as foreign by opsonin. Okay, surface receptors on phagocytes. Actin cytoskeleton rearrangement forms pseudopodia around nanoparticles. Nanoparticle enclosed in a phagosome which fuses with lysosome and degrades. Clearance mechanism but can be exploited for targeted delivery of immune cells. So suppose what happens during phagocytosis is suppose okay. So suppose this is your cell membrane. Okay, this is your cell membrane. Now you made some nanoparticles. Now these nanoparticles you've injected via IV. Now it is in circulation. So what happens when these nanoparticles comes in contact basically with the biological fluid? There is some proteins get absorbed or interact with these nanoparticles that we called as opsonization. Some charged groups, corona proteins, they could get absorbed onto it, which we call as opsonization. Now, once it has been done, what would happen next? The surface receptor uh, on phagocytose. So, what would this would be happening? So, this is a solid particle. There would be pseudopodium formation. This would be formed because of the plasma membrane forming this kind of structure, and then it will be taken in. Pinocytosis basically means external fluid, you are taking in the external fluid. And receptor mediated endocytosis, obviously there would be receptors attached on the surface of cell membrane. Different nanoparticles or nanomaterials would come and bind to these, only those particles that are specific to these receptors would bind, and then there will be internalization. So pinocytosis again is the same, trigger non-specific invagination of plasma membrane. Non-specific invagination of plasma membrane mechanism, plasma membrane folds inward. Okay, just a second. Plasma membrane folds inward, small vesicles, it folds inward. There is a formation of some kind of invagination. Now, always remember that how is it being formed. In case of phagocytosis, there is formation of the pseudopodium. Okay, and now there is this vesicle formation, invagination, inward invagination. So uptake depends mostly on local nanoparticles concentration in the extracellular medium. This is the extracellular medium. The other one is clathrin mediated endocytosis. So what happens in this? Nanoparticle surface ligands find specific cell surface receptors. Okay. If you remember in previous class, we have talked about that cancer cells have overexpression of certain receptors. Like suppose this is a cancer cell and this one's a normal cell. Okay. Now, if you're talking about transferrin receptors, so the difference that would be noticed is in case of normal cell, there would be transferrin receptors, but the expression would be low. But in case of cancer cells, there would be overexpression of these transferrin receptors. Okay, there would be overexpression, overexpression of these transferrin receptors. So in those cases, when you are suppose you have a nanoparticle, you are coating it with transferrin. Now it will go and bind to transferrin receptors. So mechanism, what is the mechanism? Ligand receptor interaction recruits Clathrins, triskelions, and lattice coat is formed. So, as you can see, suppose this is the receptor molecule and the surface of cell membrane. Now, your nanoparticle, nanomaterial, whatever it is, it comes and binds to this receptor, and now this AP2, this complex activates clathrin, which goes and binds to this particular AP2 complex, which forms clathrin coated vesicles. This one forms clathrin coated vesicles and eventually it breaks off from the surface of cell membrane and form a closed enclosed structure. And uncoating happens and finally there is fusion. So this is how clathrin mediated endocytosis works. Caveolin mediated endocytosis is more or less similar to clathrin mediated in its mode of mechanism, but there are different proteins and different molecules involved in caveolin mediated endocytosis. So what happens? What is the trigger? Why caveolin mediated endocytosis happen? The reason is interaction of nanoparticles with lipid raft domains. Okay, so there is interaction with lipid raft domains like cholesterol, sphingolipid rich microdomains, like that is all that would be present on the surface of cell membrane. I mean, on the cell membrane. Caveolin proteins form flash-shaped invaginations that is called as caveoles. Dynamin-dependent scission creates vesicle. Now, once this invagination has happened and the nanoparticles is inside, this dynamin is involved in scission. It breaks it from here and then releases it. Cargo transported to caveosome or endoplasmic reticulum Golgi, often bypassing volume. Key points: protect nanoparticles from enzymatic degradation, useful for gene therapy or peptide delivery. That they are being, these are the primary mechanisms, or I would say the mechanism that has been identified to be involved during various gene therapy or peptide delivery applications. Now the other one is macropinocytosis, which is basically it is also one of the endocytic mechanism of cellular uptake. So growth factors or oncogenic signals like RAS activation, etc., stimulates membrane ruffling. Mechanism: actin drive ruffles from large vesicles, macropinosomes, not receptor specific but bulk uptake. So there is bulk uptake of these fluid. Okay. Key points: exploited by cancer cells. Nanocarriers can preferentially target tumors. So how we can preferentially target tumor? This we have already studied in previous lecture. The next mechanism is clathrin caveolin independent pathways. trigger interaction of nanoparticles with GPI anchored proteins or flotillin rich lipid rafts. Okay, so this is also part of endocytic mechanism. So what is happening here? This one is still under study but involves small vesicles independent of classic core proteins. Cargo may traffic to recycling endosomes or bypass lysosomes, alternative entry routes for specific nanoparticles or pathogens. So suppose if it is not clathrin mediated or caveolin mediated, it could be clathrin caveolin independent pathway. One of the example is of this flotillin mediated endocytosis. Uses flotillin rich lipid wrap microdomains. This is flotillin rich lipid wrap macrodomains uh, and to enter via flotillin dependent uptake. Pegylated polymeric nanoparticles sometimes prefer this pathway. So pegylation, if you remember, I have told you that if you have this kind of nanoparticles to make it more stable in basically in aqueous solution, you coat it with PEG. You coat it with PEG. So that is what it's written here that pegylated polymeric nanoparticles like this one sometimes prefer this pathway, this protein mediated endocytosis. Micropinocytosis, what exactly is micropinocytosis? Micropinocytosis, like raft mediated uptake, lipid raft clustering triggers invaginations without clathrin caveolin. Okay. Now, nanoparticles possess charged polymeric nanoparticles and graphene oxide nanosheets. So, this is how this one is working. Now, flotillin exactly what exactly is flotillin? Because we have just now discussed flotillin mediated endocytosis, a membrane associated protein found in lipid rafts, marker of the clathrin caveolin independent pathway. So, if flotillin is present, that's a marker that clathrin caveolin independent endocytosis is happening. That whatever endocytosis is being happening, it is not because of the clathrin or caveolin. Now, helps from flotillin-rich microdomains that can invaginate and internalize cargo, including nanoparticles, pathogens, or proteins. The next one is while endocytosis. Endocytosis includes all phago, pino, CME, caveolin, pinocytosis, etc. Is the main pathway for nanoparticle entry. There are also no endocytic pathways that allow nanoparticles to enter cells. These are usually energy independent and direct translocation route. So how can direct translocation be happen? So all these that we are discussing is just via non-endocytic mechanism. So if you guys remember about, I hope if you have a biology background and even if you don't have a biology background, you would know the messenger RNA vaccines, you would know the messenger RNA vaccines that were being used by Pfizer, BioNTech, Moderna, etc. for COVID-19, basically during the time of COVID-19, if you remember. So this was the whole point. So what is happening, how they are delivering it. Suppose if you look at this particular example, okay. Suppose if you look at this particular example, just a second. So if you see, this is the SARS-CoV-2 virus, okay. This is the SARS-CoV-2 virus with the spike proteins, and in addition to that, it has its genetic material. Now you removed it and you loaded messenger RNA production according to construction plan and packaging in lipid nanoparticles. So this particular DNA was entrapped in this nanoparticles, okay, and then it was given to body cells. Now, what would body do in response to a foreign agent or foreign genetic material if it's coming? So, it would to combat it. It would start expressing antibodies. Now, these antibodies can finally fight the infection. So, that was the whole point. That was the whole, I would say, principle or essence behind these mRNA vaccines. And it was a huge craze about mRNA vaccines, especially during COVID time. Although a lot of us could not afford it, but later it was made available to everyone and we could get the vaccination. Okay, now moving forward, the next one is cellular entry of nanoparticles via non-endocytic mechanisms. The second method is nanoparticle induced pore formation. Now, how is it happening? Let us understand. Suppose there is strong electrostatic interaction between the cationic nanoparticles and negatively charged membranes. Now, suppose there is some residue which is positively charged. Okay, now you induce this residue to form nanoparticles. What would happen? It will express the free residues or the positive charge it has on its surface. Okay, now when these nanoparticles, and this is your cancer cell. So a lot of basically usually क्या होता है, these cells have negatively charged potential. A lot of portions on the cell membrane of cells is negatively charged. So strong electrostatic interaction between cationic nanoparticles and negatively charged membranes create nanopores. Now, if it would be too high, like plus 40 millivolt, plus 45, 30, so it would rupture this cell membrane and will create pores here. It will create pores here. It will be nanopores. Allows direct diffusion of nanoparticle or cargo. But if it is not at plus 40 millivolt, so usually for cell-based applications, I would say minus 15, like as per my observation and whatever experiments I have done, I would say that minus 15 millivolt to 20 millivolt is a safe window. Sorry, positive way. I'm talking about positive. Plus 15 millivolts or plus 20 millivolt तक usually they do not cause cell cytotoxicity, because what would happen is if it has positive charge on its surface, it will interact with the negative charges, and if the positive charges are too high, it will rupture the cells. It will create nanopores. Okay. Now the next one is diffusion through lipid bilayer. So a lot of small nanoparticles, like if you see this schematic of gold nanoparticles, you can see as the color is changing, the size is also changing. So this is happening because of the reducing agent that are being added to the solution. So like if the particles are very small, like 3 to 5 nanometers, they can simply get diffused through the bilayer mechanism. Very small hydrophobic nanoparticles can directly diffuse across membranes. Ultra small gold nanoparticles, 2 to 5 nanometer, can slip through membrane without vesicle formation. Some quantum dots coated with hydrophobic ligands diffuse simultaneously. Okay, so the next is cellular entry of nanoparticles via non-endocytic mechanisms. And the fourth one is transport via membrane protein ion channels. So what is the mechanism? How these membrane proteins actually help or function as protein transporters. Now, let us understand this by simply visualizing. You have a cancer cell, okay? Now, during the time when this cancer cell is growing, it would require some nutrients or like one of the major requirement is carbohydrate, because these cells are proliferating very fast, so they would need glucose. They would need iron or a lot of things. Okay, now based on the nanomaterial, the type of nanomaterial that you are making, what you can do, suppose if your cell is growing, you would require glucose, so you can simply do the glucose coating. Or you can simply do like in this example what they have done. You can see they have taken one one chemotherapeutic drug, which is cisplatin, and loaded it in GPU. What is GPU? GPU here is uh, basically they are referring to glutathione responsive biodegradable polyurethane. So glutathione, polyurethane, cisplatin with glutathione and polyurethane. They are making. So glutathione is a natural tripeptide and a major intracellular antioxidant. Many cells, especially in the brain, liver, and tumors, express glutathione transporters. So as you can see, a lot of cells expresses glutathione transporters. So suppose if we have our nanoparticle and we coat it with glutathione, we coat these nanoparticles or fabricate it in such a way that the outer layer is glutathione. These nanoparticles will come and interact with the glutathione receptors and will finally be internalized. Okay. Just a second. Now, coating nanoparticles with glutathione, just GSH, increases biocompatibility and circulation time, obviously, because cells would not consider it as a foreign and it will easily be internalized. Promotes active uptake via GSH recognizing transporters, enhances blood-brain barrier penetration. So this is the example in which they have used this and synthesized glutathione-sensitive polyurethane nanoparticles, and they have also loaded a chemotherapeutic drug which is cisplatin in it. Now coming to Trojan horse mechanism. If you guys remember in Greek mythology, there is a mention of this uh, Trojan horse. So what was Trojan horse? It was given as a gift to city of I don't remember exactly the name. There was a horse in which warriors or soldiers were hiding. So Trojan horse mechanism of drug delivery is inspired by the ancient Greek story. Drugs the warriors hidden inside something. This something was a big wooden kind of horse. The body naturally accepts the horse so that they can bypass. So why did they do? So there was a horse in which a lot of warriors um were hiding, and that horse was taken to the war site. So the warriors obviously came out and they fought the war, they opened the gate. So all those things are there. So it is a so basically what we are talking about is being deceptive. Deception is there. Okay. So nanoparticles piggyback inside biomolecules, proteins, exosome, lipoproteins, because there are some proteins that are present in the body already. Then exosomes, lipoproteins. So all these are, you know, naturally these naturally occur in cells. So if we are loading our therapeutic molecule or our drug of interest in these, it would be a good thing. Like nanoparticles piggyback inside biomolecules. So we choose those biomolecules that that can mimic the actual biomolecules present inside the cells. Example, gold or iron oxide nanoparticles coated with albumin are internalized via albumin receptors. Nanoparticles cloaked in exosomes or cell-derived vesicles evade clearance and enter recipient cells directly. So this is the example. For example, glucose-coated nanoparticles bind to glutathione receptors. Okay, transferrin conjugated nanoparticles bind to transferrin receptors. So this is how it goes. Now these are some of the sample questions again. I would request you. I'll wait for 5 to 10 seconds for you guys to attempt it. Please try attempting it. 10 more seconds. I'll wait for 10 more seconds. Please read questions carefully and I will encourage you to write the answers in the chat box. So just now we studied about the different cellular uptake mechanisms, different mechanisms with which nanoparticles get internalized inside cells. So it was basically divided into two categories. The first one was endocytic pathways, and the second one was non-endocytic pathways. So these questions are basically based on different endocytic pathways of the uh, through which internal nanoparticles gets uptaken by the cells. So let us try doing the first question. The first question says, which one of the following is the primary pathway for nanoparticle internalization in most mammalian cells? The correct answer would obviously be endocytosis. Okay. The correct answer would be endocyte. Sorry. Not endocytosis. Answer would be endocytosis. So the next question reads, clathrin-mediated endocytosis is characterized by. Okay, so what is that one identifying feature with which you can identify that this one is clathrin-mediated endocytosis? So the options were formation of cavules, no, involvement of catherine-coated pits, direct fusion with the plasma membrane, transport via ion channels. Now there could be confusion between these these basically. So obviously the answer will be involvement of clathrin coated fluids, because if you remember, this is the normal uh, surface of cell membrane. Now, when there is clathrin mediated endocytosis, what happens? This kind of some invagination forms. This is a clathrin pit. So over the time, this clathrin pit encapsulates the whole of the nanoparticle. So this is how clathrin mediated endocytosis happens. The second question is, caveole mediated endocytosis is often associated with. Okay, so the answer will be lipid wrapped regions of the membrane. Why? Cavules are small, flower shaped invaginations rich in lipids. So obviously, the bond would be strong that he has lipids. It has lipids. Uh, they are rich in lipids that mediate nanoparticle uptake. Now let us do some more questions related to, because see, you guys are already getting a lot of assignment quiz. You have the course content with you, so there's nothing that I can teach you extra, or maybe something that I'll be teaching something different from your syllabus. So whatever I'm teaching, it's basically part of your content that has been already uploaded on NPTEL website. Correct? So utilize this class more to solve sample questions, and most importantly to grab the proper theory, to grab the principles, to grab the theory behind a particular mechanism or particular phenomena, because the more you will read, more you will learn, the more you will be able to apply it in coming future, if obviously you will be going for research, if you're going for research. Now the next one is, macropinocytosis is distinct because it. Why do you consider macropinocytosis is different? So the first answer is, involves clathrin coated pits, engulfs large volume of extracellular fluid, non-specifically requires nanoparticle surface charge, only occurs in bacterial cells. The correct answer would obviously be the second one, engulfs large volume of extracellular fluid. Because what does pinocytosis mean? Break down this word. What does pinocytosis means? That it can, and then there is a word macro. So engulfs large volume. So it means that it engulfs large volumes of extracellular fluid non-specifically. The second question is, which factor does not significantly affect nanoparticle cellular uptake? So what are the features that do not affect the nanoparticle surface uptake? Now, if you remember, we have talked that nanoparticles are very small in size. Okay, so if these nanoparticles are not small in size, so obviously size will not be the option, because as the size of the nanoparticle increases, their property also increases, and then at a certain after a certain uh, limit, they will not be nanoparticles. They would be like microparticles or like that. Then shape also matters, because sometimes they are rod shaped, sometimes these nanoparticles are star shaped. Now, whenever there is a change in shape of nanoparticles from rod to sphere to star, so obviously it will depend on shape. So say shape will also not be involved, considered as a factor that does not significantly affect nanoparticle cellular uptake. The second one is surface chemistry. And the last one is light. So surface chemistry, obviously, because You will be modifying it, you will be fabricating. So this would not be the answer. So the correct answer would be ambient light because it does not have any role to play in it. So ambient light, physical and chemical properties of nanoparticle determine uptake. Light generally has no direct effect. Okay. Now let us see the interesting part. Let us come to the interesting part, which is the various methods that are being used to determine nanoparticle uptake. Now, suppose we talked about synthesis of nanoparticles, we talked about its characterization. Now, suppose you have cancer cells, you have cancer cells. Now, and you treated this cancer cell with, you carried these cancer cells with nanoparticles, and finally, suppose it has gone inside. Some would remain outside also, some would go inside. So, this is the amount which the cell has taken, is basically the nanoparticle uptake, the amount of uptake that the cell has undergone. So, how would you determine it? How would you quantify it? The first one is fluorescence microscopy or confocal laser scanning microscopy. This one. Okay. So, what happens here is nanoparticles are tagged with fluorescent dyes. So, in this case, this nanoparticle, okay, these nanoparticles would be tagged with fluorescent probes. There would be some kind of fluorescent probes that would be tagged on these nanoparticles before treating the cells with it. So nanoparticles are tagged with fluorescent dyes or quantum dots after visualization of nanoparticle localization inside cells. Confocal 3D imaging plus co-localization with endocytic markers are done. Okay, so now if you look at this image, what is happening? You can see these are the cancer cells. Okay, now this green area basically highlights a cytoplasm kind of cytoplasm of the cell. This one is nucleus. Okay, this one is nucleus. Then some other channels, whatever fluorescent agents or those things they must have used. We'll also see and understand this particular cell staining in a different slide also, the subsequent slides. So here what they are doing, they are taking the cells, then they are taking these nanoparticles basically, these nanoparticles and tagging it with the fluorescent probes. Sometimes it also happens that nanoparticles itself carries contrast agent within it. So in that case, there would be no need of tagging fluorescent probes to nanoparticle. So there would be NIR contrast agent or different fluorophores already there in the nanoparticle. So in that case, you don't have to tag it with additional fluorescent dyes. Now the other means is electron microscopy. We can prepare cell suspension, we'll treat it with cells, we'll treat it with drugs, and after it is done, after the work is done, it is processed for TEM imaging. So nanoparticle internalization by cancer cells. So they have shown that the nanoparticles that they have made are internalized inside the cells. So this is also one of the methods to determine, to check that your nanoparticle has finally uptaken the particles. Sorry, the cells have already uptaken the particles. So this is one of the methods through which you can identify that. Now, the other topic is fluorescence based imaging. Like just now here you saw that the cells are stained with different dyes. So let us first understand what are the different components of cell that we usually label using near infrared fluorescence dye. Just a second. Okay. So the first one is DAPI or HOISH. Both of them binds to minor groove of DNA. DAPI is 4,6 diamidino 2 phenylindole. It targets DNA. So as you can see in this particular cell, this blue section that you are seeing, it is basically nucleus. So if you're using only DAPI to stain cells, you will only find nuclear staining. Okay, these are nucleus, these are healthy nucleus. This is the image from control cells. Now suppose you have added nanoparticles to it that has drugs that damages DNA. So whenever there would be a DNA damage or the nucleus is getting dysmorphic, so whenever there's a DNA damage, you would be able to see it. Can you see the difference? Like, see these cells. These are control cells, okay? These are treated cells. So what has happened? We can easily detect dysmorphic nucleus. So upon ultraviolet excitation, DNA, DAPI DNA complex emits blue fluorescence. It emits blue fluorescence. Okay. Use nuclear skin for fixed and live cells. Now the second one is this mitogreen. So mitogreen, as the name suggests, is basically used to image mitochondria. Okay, a lot of time there are some drugs that causes imbalance to the mitochondrial potential or damages or causes some, um, I would say, disadvantage or to mitochondria that is basically detected by staining the cells with mitogreen. Mitogreen is a cell permeant dye that accumulates in mitochondria, largely independent of mitochondrial membrane potential. Once inside, it reacts with mitochondrial components and gives. Once it gets internalized inside the cells, it reacts with mitochondria. So this green, intense green band that you are seeing is basically because uh, it reacts with mitochondrial component and gives fluorescence. Emission 490, excitation 490, emission 516 nanometer. The other one is this phalloidin dye. See, you can obviously Google the name of these fluorescent dyes. You can read how they work. A lot of things are there. But there are some of the dyes like sometimes when you study some specific topics, when you study some important topics, a lot these a lot of examples are given, a lot of scenarios are being talked, which actually helps you get knowledge of what is currently being used. So that is why whatever examples I place here in the slide, these are the common ones. Like this particular slide, if I talk about this fluorescence based imaging of cells. So everybody, like the most common one used among all these is DAPI. Then there is uh, phalloidin and mitogreen. These are extremely expensive. So a lot of times we don't have access to such dyes through which we can do the experiment. But what happens is you should know that what are the different dyes, what are the different imaging dyes that shows fluorescence, what is the principle behind that, what is the reason that they are showing fluorescence, then which component of the cell these particular enzymes are going to and binding with. So all these are very basic questions that you should know and that you should definitely know. Now, this one, this phalloidin, okay, what does phalloidin bind with? Phalloidin binds with basically the F-actin filaments. So the target of phalloidin is F-actin filaments of the cytoskeleton. Just a second. Is anybody trying to say anything? I'll just check my audio video. Okay. I hope I'm audible and I'm visible to everyone. Okay. Let us continue. Okay. So what does phalloidin do? Phalloidin basically is used to stain the cytoskeleton of cell. Okay, it is used for cytoskeleton staining. You can see here. Now, these are the healthy cells that has been stained using pallidin. Okay, you can clearly see the cytoskeleton here. Now, these are the treated ones, the ones that you have treated with your nanoparticles. Can you see how the morphology of these cytoskeleton has changed? Okay, you can clearly see the intact, clear, crisp cytoskeleton in this case, but here it has been damaged. So all these are indicators. These imaging that you are doing, all these are indicators. These are being used as indication to test if your nanoparticles are being uptaken by the cells, and if they are uptaken by the cells, where they are going, which part they are affecting. So So all these imaging thing, nucleus, and then you take because obviously the excitation and emission for each of these channels like green, blue, red varies. So you record images individually, and then what you do, you overlay it. You stack these images one over the other, and once you do it, you'll get an something like this. So this you can do in software only, the software that you will be using for analysis for fluorescence based imaging. Now, moving forward, another example for fluorescence based imaging of cells is this live dead assay to check the proportion of live and dead cell population. Now, suppose this data is from one of my papers. Okay, so what I have done here is this one is controlled when I have not given any treatment. Then when I have given treatment, so in this case, as you can see, now let us understand what is the whole concept. So suppose there are cancer cells, okay, there are a lot of cancer cells. You made some nanoparticle and you treated these cells with it. Now, suppose if I talk about the exact experimental condition, so there would be a lot of cells, okay, in this, suppose in this well, there is a lot of cells. You added your nanoparticles here. Now, these nanoparticles would have some effect, then you will give, they would have some effect, and once the process is complete and the cells are killed. So, what next is you want to analyze how much of this, how much live cells are there. Suppose I am using green for live cells, okay? How much live cells are there because they would be live cells, sorry, they would be, all these are live. I have marked it with red, but all these are live. Now, in this case, this one, the treatment group, the one that has received treatment, it will have it will have proportion of both live and dead cells. So live dead assay is used to check the proportion of live and dead cells following nanoparticle and therapeutic treatment. So suppose here you have the cells, here you have the cells, and all the cells are live here. Then you treated these cells with the nanoparticles, and now you want to analyze what is the proportion of cells that were killed in response to these nanoparticles. So what you would do, you would stain these cells, okay? You would stain these cells, all of it. These are cells basically with, there are two dyes. The name of one of them is calci and the other one is propydium iodide. The way these, I would say, fluorescent dyes work are a little different. Like, suppose this is a case of live cell. So what would be one of the characteristic feature of live cell? the one important characteristic of live cell will be that it will have an intact membrane. Okay, because the cells are viable, healthy. These are live cells. So uh, the cell membrane will have its structural integrity. But in case of dead cell, which could be because of the stress or because of any other reason, there would be a break. There would be uh, hamper of structural integrity of this cell membrane. So what would happen? Whatever dye you would add, it will easily penetrate into the cell and bind to DNA. But in case of live cells, where we are using calcine, it goes inside cells as calcine uh, acetyl methoxy, as this dye molecule. This is the dye molecule. So I am basically trying to explain you people what is the principle behind calcine dye staining a cell red. So this particular dye, this calcine dye goes inside the cells where this part, this acetyl methoxy part is broken down by the enzyme esterase. Now this esterase enzyme is only found in this esterase enzyme is present only in live cells. It is not present here. So this esterase enzyme would remove this acetyl methoxy and this calcine in free form will give green fluorescence that you are seeing here. So in this case, all cells are live. That is why you're getting green fluorescence. So calcine acetyl methoxy is colorless. But when it is being when this acetyl methoxy is removed by esterase enzyme of the live cells, it gives green fluorescence. So it will show green color. Now, how does this propydium iodide works? So propydium iodide can penetrate and go and bind with DNA. So there is DNA intercalation. So once it goes and binds to the DNA, a red fluorescence would be obtained. But uh, since these cells are dead cells, they don't have esterase, they cannot convert it. So in that case, obviously this calcium won't come inside and bind. Now, what is calcium, which is a non-fluorescent? So in this condition, in this stage, when it is calcine acetyl methoxy, it is non-fluorescent. But as soon as it becomes calcine, it gives green fluorescence. Then it is cell permeable dye. Now calcine is a cell permeable dye. It can penetrate cell. That is converted by intracellular esterases in live cells into fluorescent calcine emitting green fluorescence. Propydium iodide. What is the nature of propydium iodide that we just discussed? A membrane impermeable dye that enters dead or membrane compromised cells, binds DNA, and emits red fluorescence. So this one is emitting red fluorescence. What is the principle behind live dead assay? Whenever you are working, whenever you're studying about the anti-cancer properties and all those things, you need to start your work from cell-based experiments. Suppose if I talk about the correct flow, first thing that you need to do is synthesize nanoparticles. Once you have your nano construct or nanoparticles ready, the second thing that you do is do its characterization that we have also already discussed in the second class. That what are the different characterization techniques, MVLS data, UV, Flurry, a lot of things are there. Then the next step will be you will check the efficacy of your nanoparticles on cancer cells, on these cancer cells in vitro condition. So that would be your second part, that all of it needs to be checked in vitro conditions, that what effect they are causing on cells. So now you have a nanoparticle, you want to check its anti-cancer properties, or mainly I should say in simple language that how much capable these nanoparticles are in killing cancer cells. So in order to get a clear idea, I should know that if there are this number of cells that I have taken for my experiments, then after the treatment with nanoparticles, how much cells are still remaining? Okay, so that is why it's important to know the exact proportion of both live cells and dead cells, so green and red, both. Okay, now this is done. Let us discuss another thing. Okay, some uh, Is someone trying to say anything?
Ma'am, yes, ma'am. Ma'am, previously, so when you're doing that in vitro, that usually for analysis, you have to see whether it's viable or not. So we are going to add both of the compounds, calcine AM and the potassium iodide, the PI. Both you are going to add, ma'am, in the
Yes,
this thing.
we have to co-stain. So there is a
Propylene, ma'am.
term, they will be co-staining. That's a very good question. So there is co-staining. You have to stain cells. Suppose these are the cells. You have to add both calcium and PI. So there are co-staining. There's a term called as co-staining. Even in the previous image that I showed you, so these cells are co-stained with both calcine and PI. This is from one of my papers, but here also there is also co-staining. The cells has been co-stained with all three of the dyes, DAPI, this mitogreen, and this phalloidin, this one. So we have to co-stain. We have to add both the dyes together.
Just like when we do gram positive and gram negative, we will do both staining. So same way we are doing here.
Yeah, correct. Because we want to know
Okay.
the exact percentage, how many live cells are there, and based on the therapeutic efficacy of the nanoparticle that we have prepared, what is the potential of those nanoparticles in actually killing the cells or maybe inducing stress. Now, there are two things. Now, in the subsequent slides, we'll be studying about the different in vitro methods that are used for checking the anti-cancer properties of nanoparticles. And the first one in that would be cell viability assay. That after treatment with nanoparticles, if the percentage cell viability, or I would say the percentage of live cells is getting reduced. Now, at that point, I will compare the functioning of, you know, two things. Now, sometimes what happens is when you are using live dead assay to estimate the efficacy, the therapeutic efficacy, and when you compare this result with MTT assay or any cell viability assay in which you are computing the exact percentage of cells that are dying. So the difference arises because here the principle is different. Whenever there is, uh, suppose the loss of membrane integrity would happen even in cases when there would be, mitochondria, nucleus, the other organelles would be there. So even in case of stress, what happens is the membrane permeability gets compromised, so this PI would seep in. But in case of MTT assay, which is basically based on the mitochondrial enzyme succinyl hydrogenase activity, so even when the cell membrane integrity is compromised, there could still be mitochondria and succinyl dehydrogenase that is used to estimate percentage cell viability or the percentage of live cells. So whenever you're comparing, if you have read some research papers, you would be seeing that there are different experiments being done to prove a single point. Suppose I am saying that my nanoparticles are killing cells at this particular concentration, and the percentage of cells left is 20% only when I am doing this live dead assay. But in case of MTT assay, there is only it is 30%. So they would never be like it would never overlap. The sensitivity, the principle of each of the technique, of each of the method that we use in vitro studies are different. And because the principle, the working principle, the sensitivity is different, the data would not overlap. But at least you will have an idea that, not idea, but you can, with all those data, all those separate data, you can use it as a corroborative data. That one particular data set comes in support of the other experiment. So, since the working principle differs, the workflow differs, their sensitivity and would also differ, obviously. So, for this one, the question that was basically asked was, you know, if calcium PI needs to be added together, yes, we'll be doing co-staining. So, the next thing that is used to determine the nanoparticle uptake is this technique, which is called as flow cytometry. Now, flow cytometry again is a very, I would say, important technique and very potential technique to do this work. So, cells are incubated with fluorescently labeled nanoparticles. Flow cytometer measures the fluorescence intensity per cell. Now do remember this. Okay, and how is it happening? Because of the hydrodynamic focusing, which aligns cells in a single file. Sorry, correlating with nanoparticle uptake. Advantages, it is rapid. Statistical analysis of thousands of cells can be done. Now, this is the place where why I was repeatedly stressing on sensitivity. Now compare what you're doing in this. If you're doing the same analysis, okay, if you're doing the same uptake analysis with flow cytometry, and same if you are doing with fluorescence microscopy. Now, in this particular field of view, suppose this was your complete cell sample and you are imaging this part. Okay, this is your field of view. This is your field of view that you are visualizing. This is the field of view. Now, what would happen is it will not be statistically significant to if we are predicting the efficacy based on this only one field of view. But in case of facts, the sensitivity, it's much more sensitive. Why? Because in this case, we are kind of analyzing thousands of cells in one go. So flow cytometry is a powerful technique to analyze physical and chemical properties of individual cells as they flow in a fluid stream through a laser beam. So this is the sample. Your samples are usually stained with some of the dyes, like sometimes annexin PI for live death staining, and for then annexin VPI for apoptosis, and then flow cytometry sends cells in a stream one by one, because of the hydrodynamic focusing. Then it measure light scatter plus fluorescence based on the labels that we have added, and then collect data for thousands of cells. Let us understand it in a bit detail. So, one more thing, always remember that we use sheath fluid to make the cells flow through it. Sheath fluid is important, and the flow rate and everything, gating, all those things are decided. Those are the complex things, but let us first understand only the principle. Now, the sample is passed through it. There is a nozzle, and your sample would be irradiated by this laser light source. So, each of the cell would behave differently depending on uh, you know what kind of dyes are there, if those cells have undergone apoptosis or not, and if the binding with some antibodies have happened or not. So each one of it will give a different signal. Detection of fluorescence emitted by stained cells. Now the cells which will not be stained, detection of forward and side scatter light from all cells. So based on all these data that is collected, a graph is plotted that I will show you in the next slide. But let us first understand the principle of flow cytometry. So the first, fluidic cell suspension flow. Cells or nanoparticles are suspended in a liquid. They pass single file through a narrow nozzle using sheath fluid. I just now explained, they use sheath fluid and they pass the sample. Each cell passes through the laser beam, light scattering occurs. So as each of these cells, this one, this one, as it passes through laser, light scattering would happen. It which tells about size and granularity. So if there is forward scatter, whatever is the proportion of forward scatter, it is proportional to cell size. And side scatter, if there is side scattering happening more, it is related to internal complexity or granules. Now, if the cells that we are supplying here are tagged with fluorescent probes like annexin V and PI, which is the commonly used one, uh, if the cell nanoparticle is labeled with fluorescent dyes, antibodies, or autofluorescent nanoparticles, detectors capture the emission at specific wavelengths. So the detectors would capture emission at specific wavelengths. Then detectors, usually photodiodes or PMTs होते हैं, which convert light signals into electronic signals. Data is digitized and analyzed as histograms or dot plots. What are histograms or dot plots? So whenever you're looking for this FACS spectra, you would find something like this. It will be divided into four quadrants: quadrant 1, 2, 3, or 4. What does all these quadrant means? So as you can see that on the x-axis there is annexin V-FITC, and on the y-axis there is propydium iodide. Annexin V-FITC stains live cells. Propydium iodide stains dead cells. Okay. Now, based on that, let us understand what does it mean. So, the quadrant 3, this one, both. So, whenever this dot plot, the cells are falling in this plot, it suggests live cells. Here, Q4, early apoptotic cells, means the proportion of cells that have entered early apoptosis and may die. Q2, late apoptotic or secondary necrotic. So in this case, these are late apoptosis stages and some necrotic cells would also be there. And Q1 basically are necrotic cells. So suppose if you are claiming that your nanoparticle, that your drug loaded nanoparticle, suppose you have a nanoparticles in which you have loaded a certain drug which causes apoptosis. Okay, these nanoparticles causes apoptosis. So what should be there? Your cells should lie either in this or some of portion can lie in this quadrant for apoptosis to prove that these nanoparticles are causing apoptosis and not causing necrosis, which is not considered good. Now, this one is a scatter plot for distinguishing cell types by size and granularity. X-axis, forward scatter, which tells the size. Y-axis, which is side scatter, which tells about internal complexity or granules. So this is how you study FACS data. So these are some of the technicalities. If you read some research papers, if you, are very much into science and you like it, this is how the FACS data are studied. So you can use this information to further extract information from other research papers who might have done FACS. So let us do some quick uh, questions based on FACS. So I'll wait for 10 to 15 seconds. Please try attempting these questions. Okay, try attempting these questions. Okay, so just now we studied about FACS, so that is fluorescence activated cell sorting. So what do you think? What is the primary purpose of FACS in nanoparticle studies? Let us read the options. The first one is measure nanoparticle size. The first one is measure nanoparticle size, quantify cellular uptake, and sort cells based on fluorescence. Determine nanoparticle solubility, visualize nanoparticle under a microscope. The answer is very clear, obviously the B1. For what we do FACS, we use it to quantify cellular uptake and sort cells based on fluorescence. This is one of the applications. The other question reads, which type of dye is commonly used for tracking nanoparticle uptake in FACS? So what are the dyes that are used? Obviously, fluorescent based dyes are used. Okay, now moving forward. In fluorescence based staining, okay, the question is asking about fluorescence based staining. What does a high mean fluorescence intensity indicate? If you don't tell me, mean fluorescence intensity basically is the average of the fluorescent signals that you have observed based on the fluorescent signal you have observed for a particular dye or something used to indicate. So the correct option would be, let us read the options one by one. The first one is cell death, higher nanoparticle uptake, lower nanoparticle uptake, no correlation with uptake. So obviously, if the mean fluorescent intensity is high, obviously it denotes that the uptake of nanoparticles are high. That is something very obvious. Suppose you have nanoparticles and you have tagged it with fluorescent probes. Okay, you have tagged it with fluorescent probes. Now, if it is showing, if while doing imaging, when you are doing imaging, these are cells here, and all this, it's showing high mean fluorescence intensity, means the signals are very high. It means a lot of the nanoparticles are being these nanoparticles are being uptaken by the cells. So more the nanoparticle uptake, more would be the signal coming out from the cells, so more would be the fluorescence intensity, mean fluorescence intensity. Second question is propadium iodide is used in fax primarily too. Just now we have seen calcine with Pi and XNV plus Pi. So I think you all must be knowing this is L. Calcine stains live cells. So obviously propadium iodide would stain dead cells. Always remember. Now, question number 5. Which of the following can affect fluorescence-based nanoparticle uptake measurements? I'll wait for 10 seconds. Try attempting these two questions. So, in the question, you have been asked that when you are, sorry, so the question that is being asked is which of the following can affect fluorescence-based nanoparticle uptake measurements? Which of these following options, A, B, C, D, can affect fluorescence-based nanoparticle uptake measurement? The options are nanoparticle size and surface charge. Obviously, it will affect fluorescence-based nanoparticle measurement because if it would be of very large size, it could have sense. Then ambient temperature, shape of the microscope stage, color of the culture plate. No, it does not happen. The correct answer would be the A1, nanoparticle size and surface charge. Because suppose that is what I have been telling you guys. This is a cancer cell. Okay. Now, if the particles are very small, it would easily be internalized. Okay. It will easily be internalized. But if the particle is very large, it will not reach till here, it will not be able to internalize. Other aspect is surface charge. Now, if your nanoparticles are positively charged, if they carry a positive charge on their surface, what would happen if they carry a positive charge on their surface? There would be weak electrostatic interaction. It will react with the uh, negative charge of the plasma membrane. But if your nanoparticles also carry negative charge, then the uptake would be different. So that is why size and surface charge does play a role in nanoparticle uptake and eventually fluorescence-based uptake measurement. The second question is which fluorescence method allows visualization of nanoparticles inside specific cellular compartments? So you have to tell me which fluorescence method allows visualization of nanoparticles inside specific cellular components. So obviously the answer would be confocal microscopy because in flow cytometry you are collecting the signals and doing the dot plot. You are not able to visualize from your own eyes. But in confocal microscopy, what is happening? You can see like see this one. You can see these are the confocal microscope images. You can see nucleus. You can see this mitotracker tagged. I mean mitochondria, the portions where mitochondria is there. Then the red phalloidin, which is used for staining, it has stained cytoskeleton. So, which fluorescence microscopy method allows visualization of nanoparticles and specific cellular components? Obviously, it would be confocal microscopy. Moving forward, yeah, this one is the other method. Like we were discussing different methods that are used for estimating the cellular uptake or estimating the uptake of nanoparticles by cells. So, The other method is spectroscopic or analytical methods, inductively coupled plasma, mass spectrometry. These are some of the instruments that you can use to predict I mean, measures elemental content like gold in gold nanoparticles, iron in spions, highly quantitative and sensitive, or atomic absorption spectroscopy, which is similar to ICP-MS, and it can also be quantified using UV visible spectroscopy or fluorescence spectroscopy. Other method for doing that is radioactive or isotope labeling. So you have your nanoparticles and you are tagging it with what? You are tagging it with radioactive isotope labeling, radioactive isotopes. Example MTC, copper 64. So uptake measured using scintillation counting or PET SPECT imaging. Because once you have added radioactive isotopes, once you have tagged radioactive isotopes to your nanoparticles, these nanoconstructs have now become as a contrast agent which can be used for positron emission tomography and this SPECT imaging. Advantages: very sensitive in vitro and in vivo. Limitation: what is the limitation? It requires handling of radioactivity. So you have to be very careful when you're working here because nobody wants to get exposed. This is one of the examples. So this is radio labeled nanoparticles. Okay, these are radio labeled. You have already added the radioactive isotope. Then trapping radionucleotide into the nanoparticles. Here you have trapped radionucleotide inside the nanoparticles. Then you have added chelator-free surface labeling, chelator-based surface labeling. Once you have done that, you can do the SPECT imaging or PET imaging. Now, another approach is biological molecular approaches, co-localization with endocytic markers like clathrin, caveolin, wrap proteins used to determine uptake route, or use of endocytosis inhibitors like all these things that are mentioned. Another very interesting way to detect the uptake of nanoparticles in cells or for other purposes, we use label-free techniques. What does label-free techniques means? Means here you are not using any fluorescence agent. Okay, no fluorophore, no tagging of any contrast agent or fluorescence contrast agents. So one of the examples for this is Raman spectroscopy or surface enhanced Raman scattering, X-ray fluorescence microscopy, and mass cytometry. So Raman spectroscopy detects nanoparticles like gold nanoparticles without labels, so you don't have to put labels. So this is one of the papers in which what they have done is they have all those imaging. These are Raman based imaging. So they identify that at which place which component is present and at what intensity based on this spectra. So you can definitely read it in bit detail if you are interested. Okay. Now, coming to these methods, in vitro methods to study anti-cancer properties of nanomaterials. Why is it important? Because once you have synthesized nanomaterials and you want its intended application against cancer, so you have to have some set of experiments. So these are the set of experiments that are performed. These are the set of experiments that are performed in vitro to validate the anti-cancer properties of any nanomaterial or nanoparticle. So the first one is cell viability assay. Okay, just a second. The first one is cell viability assay. Live dead staining I have already explained you people, and rest of the things I would apoptosis I have explained like an XNPI and all those things facts. Rest of the methodologies I'll explain you one by one. So let us start. The first one is cell viability assay. What does cell viability assay mean? This assay is based on the ability of live cells to maintain metabolic activity. This is what I was telling you when I was explaining live dead assay. So here the term metabolic activity is used. Now, in case of MTT assay, suppose this is a live cell and now suppose it's dead, this cell is dead, the nucleus is disrupted and all those things. So even in this condition what could happen is because of the compromised plasma membrane, when you are doing live dead assay, the PI can penetrate here because it is obviously the membrane integrity would be compromised when the cells are dying because of the stress, which would be one of the reasons. But even after it dying, it could still have some metabolic activity left in it, some enzymes, some molecules. Okay, it could still have some metabolic activity left. So this cell viability assay basically measures the metabolic activity, the remaining metabolic activity. Obviously, there won't be much difference. The difference could be 10 to 15% or something like that when you're comparing the data. But what the only thing that I'm trying to explain you people about is the sensitivity, the working principle, and how things are being done differs from one technique to the other. So always keep in mind while analyzing the data. Cell viability assays are based on the ability of live cells to maintain metabolic activity, membrane integrity, or enzymatic function. Common methods include like MTT, XTT, WST, then Alamar blue, Trypan blue exclusion. So all these techniques are there. So we'll discuss about MTT basically because it's the most common one. It relies on reduction of tetrazolium salt by mitochondrial enzymes. So the live cells will have mitochondrial enzymes which will reduce the tetrazolium salt that we add in form of MTT. And this will be happening in viable cells. But in case of dead cells, what would happen? There won't be enough mitochondrial enzyme succinyl hydrogenase that can, you know, uh, reduce it. So MTT assay measures cell viability based on the reduction of yellow MTT dye to purple formosine crystals by mitochondrial enzymes in metabolic active states. So the live cells would show uh, like kind of dark purple color, and the dead cells would show light purple color. So basically, we measure the absorbance at 570 nanometer. Okay, we measure the absorbance at 570 nanometer. We measure absorbance of this one. Like once we have added the MTT, this one had our samples or basically cells. So this is the whole workflow, what is being done. So this is one of the example of cell viability assay, how we are doing it. How is it done? Suppose you have made nanoparticles of different concentration. Suppose there are five concentrations. एक, दो, तीन, चार, पाँच, five different concentrations. So you added five different concentration of nano. Suppose this is the plate. You have added five different concentrations in triplicates to this plate. Okay, two, three, one, two, three. Now once you have done it, you will incubate these nanoparticles with the cells, and after the incubation time is complete, you will add MTT acid. Then incubate the plate for 1 hour. So here it is written 1 hour. Usually we do it for 3 hours, 3 to 4 hours. Okay. Now once you have incubated your nanoparticles treated cell with MTT, you'll remove it and add 150 microliter Pulvel MTT solvent plus 3 blanks. So here they are adding it in this step. Once you have done that, once the incubation is complete, you will take it out and add DMSO to break the formosine crystals. And once the formosine crystals are broken down, they will give out purple color. So the live cells would be would show highest intensity, and dead cells will show the least intensity of absorbance when you will measure it at 570 nanometers. So this is how it is being done. Now again, the other thing was live death staining. I have already explained you live death staining, that live cells fluoresce green because of calcine staining and dead cells fluoresce red, allowing simultaneous assessment of cell viability. Then apoptosis assay I have also told. Although in addition to this facts-based data, you can do it via fluorescence imaging as well. Apoptosis assay you can do via fluorescence imaging as well. There is apoptosis kit that comes that will basically marks with some of the apoptotic markers maybe. So there is apoptosis kit in the market that you can order, and you can stain cells and do the fluorescence imaging. So next is reactive oxygen species generation. So whenever the cells are dying, or I should say that whenever there is stress in the cells by some mechanism, maybe because of drug, because of laser exposure, or any of the reasons, whenever there is some kind of stress or damage, the cells, or maybe there are some nanoparticles that triggers the formation of reactive oxygen species. Okay, so these reactive oxygen species, because of the oxidative stress, obviously, uh, leads to DNA damage, DNA strand breakage, chromosomal fragmentation, and gene mutations, and ultimately leads to cell death. So, if we are looking that if our nanoparticles are capable of triggering, sorry, are capable of triggering apoptosis. If they have initiated apoptosis, how can we check? By looking for reactive oxygen species generation. So Ross assays are crucial for evaluating oxidative stress in cells. Some nanoparticles during the therapy may induce this oxidative stress to kill cells, which play a role in nanoparticle-induced cytotoxicity and anti-cancer activity. Measuring reactive oxygen species. So, but how will you measure this reactive oxygen species? So that's what. For that, we use this particular dye. This is Ross detecting dye. Okay. Measuring Ross helps understand whether nanoparticles trigger apoptosis, DNA damage, or other stress related pathways, thereby providing insight into their therapeutic potential and safety. So, how is it done? How do you estimate reactive oxygen species generation? So, this DCFDA, which is 2,7-dichlorofluoroocene diacetate, is a non-fluorescent dye. If you remember, we also talked about calcium. So, till the time it was calcium acetyl methoxy, it was non-fluorescent. But as soon as the acetyl methoxy was removed from calcium by esterase enzyme, it became fluorescent. So, similar thing is happening here. So, what is happening is it's a cell permeable dye that is deacetylated by esterase. So, again, the live cells which are undergoing just entering the stage of apoptosis, they are under because of the stress. So these esterase enzyme will deacetylate. It will deacetylate DCFDA inside cells, and upon oxidation by reactive oxygen species is converted. And once it reacts, combines with ROS, it is converted into fluorescent DCF, allowing quantification of intracellular ROS levels. So based on this green signal generated in these cells, you can quantify the intracellular reactive oxygen species level, that how much reactive oxygen species is being produced inside cells following treatment. Now the other technique is this clonogenic assay. What is clonogenic assay? Now, one thing you guys would have also heard that whenever we're talking about tumors, okay, or not just tumors, whenever we are talking about cancer as a whole. So you guys would have heard that okay, there is again reoccurrence of cancer even after therapy. Uh, the cancer is again developed and all those things. So whenever we are doing these things at in vitro level, whenever we are doing at in vitro level, how do we check it? We check it via clonogenic assay. So what does clonogenic assay measure? It measures the ability of a single cell to survive and proliferate into a colony after treatment. So suppose there was a colony of cells that you treated with nanoparticles. Okay, there were cells in it. Now once you have treated, a lot of them would die. Okay, these cells will die. Now once everything is removed and all those things are there, it predicts, this clonogenic assay predicts that if there is single colony left in the egg, what is the potential of this single colony to proliferate after treatment? So this is the case of control. In case of control, obviously this is no treatment group. no treatment has been given, so obviously you can see different colonies are formed. But in case of treatment groups, as you can see, there's uh, the concentration of the treatment material used is increased, the number of colonies is getting decreased. Means as we increase the therapeutic dose to kill the cancer cells, the chances of it returning and forming colonies again and proliferating and growing would be less. So it assesses the long-term cytotoxicity and anti-cancer potential of treatments by evaluating reproductive cell death and tumor forming capacity. So as you can see, this is the highest concentration. In this case, there is no um, colonies. There's very less colonies in compared to control coming for this one, which shows that it has a great potential. It has a great therapeutic efficacy. Now another technique that is used is the scratch assay. It is again the same thing. So what we do, we grow cells. As you can see here, the cells are grown. Then using something, a scratch is made. Okay, and this distance is kept constant. So use some kind of material that creates a consistent scratch so that the distance is same. So once you have scratched all the cells, so what kind of behavior is expected is that, suppose these are the control cells in which we have made the scratch. Okay, these are the control cells. Now there were cells all around and everything, and control cells obviously will not receive any treatment. So over the course of time, after some time, what would happen is this scratch, this distance would be reduced because cells will start growing here as well. So this will reduce, and after some time this scratch will even be merged. So there would be cells growing everywhere, even, you know. So means this means the cells have migrated because these are not killed. But in case of treatment groups, okay, where the cells have received treatment, what would happen? Suppose these are cells, you made a scratch, there are cells here. Now once you have given treatment, then after T time, this gap, this scratch would be like, it would either not move. Sorry, this has been made incorrectly. But what I wanted to say is, if you'll compare the distance, this one would be low, but this one would still be high because the cells which has already gone under stress or maybe killed will not continue and grow in this scratch space. So this is the principle of scratch assay. A scratch is created in a confluent cancer cell monolayer and the ability of cells to migrate. See, as the time is increasing, this wide gap is becoming narrower. So this is no treatment group. So in no treatment group, the scratch or gap is almost closing, as you can see. But in case of 1 minute, these are the treatment groups. Okay. So these are the treatment groups. So as you can see, as the time is increasing, this gap, if you compare that this is the highest concentration or for longest time we have compared that, in comparison to the control, it is less. So this is the idea of migration and invasion assay, or what we call as scratch assay. Now another thing is how, if we want to take, those were the things when we were checking the anti-cancer properties in in vitro conditions. Now if we want to check the antibacterial properties, what are some of the experiments or what are some of the methods that we follow? So one is this broad dilution method in which we determine the minimal inhibitory concentration. That what is the exact concentration of the nanoparticle that is needed to inhibit the growth of a certain number of bacterial cells, or I mean whatever was your initial concentration of cells, bacterial cells basically. So the first step is obtain isolated colonies of bacterial strain to test. Okay, then combine 4 to 5 colonies and culture overnight in rich media broth. Means you have done the overnight culture, and then you made dilutions and added different concentration of antibiotics this antibiotic also to test. Fourth, plate alicot of growth, matlab like this one, plate one colony from this one, plate one colony from this one, to see the number of colonies that are being there. After overnight incubation, check cultures for growth. The MIC is the lowest concentration of antibiotic that prevents visible growth. So out of all these concentration, the lowest concentration that prevents visible growth would be selected. So this MIC, minimum inhibitory control, varies from one nanoparticle to the other. So this is one of the methods, which is broad dilution method. So bacteria are exposed to different concentration of nanomaterials in liquid med or antibiotic in liquid growth medium, and the lowest concentration that inhibits visible growth is the minimum inhibitory concentration. Subculturing from these tubes helps determine minimum bactericidal concentration. Now another method is this agar diffusion assay. So what is being done here? So first of all, you grow bacteria and identify 2-3 colonies and keep it in shaker incubator for overnight. Then the inoculum density is standardized using McFarland standard. Now once you have obtained the bacteria, you take a petri plate, and this is kind of earbud kind of thing, and you take a little amount of culture and you spread it through the whole plate. You spread it like this. Okay. Matlab, I mean, you start from here and doing it, you cover whole of it. Once you have done, you dug out small wells. So as you know that bacteria, this is made of agar. So if we are removing this much of agar, it would create an empty place, or I would say a well-like structure. Now you put your samples or nanomaterials or whatever you have in these wells, and then you see the zone of inhibition that has been formed. So suppose these are the dugout wells. If you put your samples here, then and leave it in shaker incubator overnight, you will see that the bacteria would develop like this from here. There would be some space here that we call as zone of inhibition. This zone of inhibition would vary depending on the concentration of nanomaterial. So this zone of inhibition would vary depending on the concentration of nanomaterial, what kind of nanomaterial it is, whether it is capable of combating antibacterial, I mean, whether it is whether it can address antibacterial, it has antibacterial properties or not. So these diameter wells are made and your samples are put and then it is left. So this zone of inhibition is calculated. So let us read this. In this method, the bacterial culture is spread evenly on an agar plate, and nanomaterials are applied either on sterile paper disc or into wells punched in the well diffusion. As the nanomaterial diffuses into the agar, it inhibits bacterial growth around the application site. The zone of inhibition is measured and its size reflects the antibacterial effectiveness of the material. So suppose this was the well in which you filled your nanomaterial sample. Now, over the course of time, this nanomaterial would fuse to the agarose well because it has agarose. So it will fuse into the agarose coating, and up to the distance where it diffuses, it will not allow bacteria to grow. So bacteria will not grow in this region. It will grow here. So this is the zone of inhibition that we use to calculate the MIC. So how do we, as you can see here, see this plate. Here you put your samples. This is the zone of inhibition. Like for high concentration, the obviously the spread would be more. It will spread to longer distance. So zone of inhibition is large. Visible zones which may be sensitive, resistant, or increased exposure need to compare breakpoints to bacteria growing right up to the disc. Here it has grown here only. Maybe the concentration of the nanomaterial or antibiotic would be low. So how do you calculate MIC? Measure the diameter of the inhibition zones. Plot zone diameter on the y-axis. So the zone diameter is plotted on the y-axis, and concentration diameter is plotted on the y-axis, and concentration is plotted on the x-axis. So based on this, a calibration curve is plotted. The MIC corresponds to the lowest concentration that produces a measurable inhibition zone. So whatever minimum uh, concentration an diameter, inhibition zone is seen, we call it as minimal inhibitory concentration. So these are some of the sample questions. I'll just wait for 10 seconds for you guys to attempt it. Try attempting these questions. Okay, so let us discuss the answers one by one. The MTT assay, as we have just now studied it, the answer would be it measures the metabolic activity of viable cells based on the presence of succinyl hydrogenase, which is a mitochondrial enzyme, which assay evaluates the long-term survival and proliferative potential of cancer cells. Obviously, the clonogenic assay because it measures that if it measures reproductive cell death after treatment. That what would happen, for how long it can be controlled, or what is the potential. DCFDS staining is primarily used to detect, obviously, reactive oxygen species. Now, some more questions related to the antibacterial, in vitro antibacterial methods. We just now read about the broad dilution method, how different dilutions are made, and the MIC is calculated. So broad dilution identifies minimal inhibitory and bactericidal concentration. So MIC is minimal inhibitory concentration and this is minimal bactericidal concentration. This one tells at which concentration it gets inhibited. All those things, and it gets killed. Which assay involves measuring a clear zone around nanometer? Obviously, we just now studied about agar diffusion assay in which see this inhibition zone. Then colony forming unit counting is used too. Obviously, it counts viable bacterial colonies after treatment because whenever you are doing any experiment, you need to start with some, you need to know that what was the colony forming units, how much bacteria or how much bacterial cells were there initially when you started the experiment, and that has to be kept constant so as to avoid discrepancies in the data. So this is how colonies are calculated. Like it has 420 colonies, 43, 51, 20 colonies. How do you count a colony? A CFU represents a single viable microbial cell or a small cluster capable of growing into a visible colony on an agar plate. After treatment with nanoparticles, bacteria are plated and incubated. The number of colonies formed reflects the number of surviving viable cells. So based on this, you can predict that what could be the effectiveness of the nanoparticles in killing bacterial cells. Significance: CFU counting is a standard method to quantify antibacterial activity and evaluate the killing of inhibitory effect of nanomaterials. Okay, to calculate bacteria, this is the formula. Like CFU is equal to number of colonies that you can count into dilution factor. What was the dilution factor you used? Like if you have 63 colonies on a plate from a 10 to the power minus 5 dilution and you plated zero point 0.1 ml, that is 100 microliter, your calculation would be this. This would be the CFU. So this is the formula for CFU calculation, and whenever you're starting experiment, you need to keep the CFU for all the groups same, experimental groups same. Then this is the last uh, topic from your this week's content, which is nanotoxicology. So nanotoxicology is basically the branch of toxicology that studies the safety, interaction, and adverse effect of nanomaterials on living systems and environment. Now, why is it important? Because nanoparticles have unique properties. They can cross biological barriers, enter cells, and potentially cause oxidative stress. Like they could be, obviously, we are using it for cancer therapy, for bactericidal effects, and all those things, but it can also sometimes damage the normal cells. It can cause inflammation, genotoxicity, or organ damage. So, long term cytotoxicity of nanoparticles shall be known. So these are the different things that you can read it from here. Then one important experiment that I wanted to teach you was this hemolysis assay. It is one of the, I would say, it is one of the methods to check hemocompatibility. Suppose you are injecting your nanoparticles via IV route. Now, if your nanoparticles, you need to check by hemolysis, you check that the nanoparticles that you are injecting via IV is not causing RBC rupture. Okay, because your nanoparticles would come in contact with blood. So if so, in this, what we do, it measures rupture of red blood cells. Because if your nanoparticles is causing RBC rupture, means it is not safe. It is not biocompatible, and it is this hemolysis assay. How does this hemolysis assay works? It measures the rupture of red blood cells. So what we collect blood, clean it, and collect an RBC suspension. Now we mix this RBC suspension with the nanomaterials of our choice for some time. RBC were nanomaterial. Now, if this nanomaterials is, capable of causing hemolysis and it is not safe, what it would do, it would cause hemolysis and a red fluid kind of, supernatant would be there. But if there is no hemolysis and when we centrifuge after centrifuge it, we'll be collecting a pellet of intact red blood cells, and there would be no hemoglobin in the supernatant. But in case of if it has caused hemolysis, we can detect hemoglobin in the supernatant. Now we measure the amount of hemoglobin using spectrophotometer. Okay, we measure the amount of hemoglobin by recording absorbance of hemoglobin via spectrophotometer, and we predict. So if the percentage hemolysis is less than 5%, means if it is less than 5% means your nanoparticles are safe. Okay. So hemoglobin released, quantified spectrophotometrically, assesses blood compatibility and safety of nanoparticles for biomedical use. Now some last set of questions. The first one is nanotoxicology is primarily concerned with obviously how nanoparticles interact with cells because it is very important. Until and unless you know that it is not causing toxicity to the cells, it is not leading to RBC rupture, you cannot use it for the therapeutic purposes. So nanotoxicology is very important. Now, which property of nanoparticles often contributes to their unique toxic effects? Obviously, their small size and large surface area. Because if the nanoparticles have small size, they have large surface to volume ratio, and because of this it will present large reactivity, okay, to penetrate, and it will penetrate cells and interact strongly with biomolecules. Now one common mechanism of nanoparticle induced toxicity is obviously reactive oxygen species production because it causes oxidative stress, and whenever there is oxidative stress there would be reactive oxygen species production. Hemolysis assay in nanotoxicology is used to assess it evaluates whether nanoparticles damage red blood cells releasing hemoglobin or not. Then, which factors most influence nanoparticle toxicity? Obviously, the first one because size, shape, surface charge, dose, all of these factors influence the cytotoxicity caused by nanoparticle. The next question is why is nanotoxicology important in biomedical applications? Obviously, to ensure safety and biocompatibility in humans because we cannot use a toxic nanoparticle or nanomaterial for therapeutic application. So that is why it is important to uh, evaluate the toxicological profile of the nanoparticle or nanotherapeutic that you are making.
Hello,
Yes,
am I audible, ma'am?
yes, you're audible.
Ma'am, it's a wonderful session, ma'am. Thank you so much.
Thank you.
Very
So
well organized, very useful for students.
thank you very much. So this
Thank you.
YouTube video will also be uploaded, and apart from
Yes,
this, you have three more videos as well. So if in case you guys have any doubt related to anything, you can write down your doubts in the comment section. So the time that is allotted to us, 2 hours, is very less. So I try to cover a lot of topics, but sometimes we might miss something. But if you still feel like that there is something that needs to be addressed, uh, uh, one of you can write in the comment section. I would address to it.
Thank you so much, ma'am.
Thank
Nice
you.
of you.
Thank you, everyone.